Hey y'all, we're back and we're going to talk a little bit more about inheritance here. We're going to talk about the Punnett square. Okay, some people hate Punnett squares. Some teachers don't like to teach the Punnett square. I don't know why. There are other ways of factoring through this, but the Punnett square is the easiest way to do this. Alright, and so on the next part of kind of the assignment we did in class, it's going to give you some information and you have to basically cipher through the information and figure out some probabilities. Okay. Now, a few things about the Punnett square. We draw it like this if we're doing a two allele cross. So in this case, the information given to you on the paper says that round seeds in pea plants, round seeds are dominant to wrinkled. All right. So round is dominant to wrinkled. So we will use big R for our round trait, and we will use little r for our wrinkled allele. Okay, so big R for round allele, little r for wrinkled allele. Now in the first cross, I think it gives you a cross like, it tells you big R, little r individual times crossed with a little r individual. Okay, so basically, let's spell this out for ourselves. It tells us what is dominant, remember? So this would be a round heterozygous individual. This would be a wrinkled homozygous recessive individual. When I put these in a Punnett square, I put this individual at the top. It doesn't really matter, but that's what we'll say. So I have my big R, little r individual at the top. I put one letter per column. Basically, what this means is, remember, you have two copies of each gene. Homologous chromosomes, one copy on each gene. So basically, let's say that the gene for round or wrinkled is located on chromosome number 2. So here's chromosome number 2. Okay. This individual on chromosome number 2 would have the genotype. Here's the gene, we'll say, big R, one copy of big R, one copy of little r. Okay, this individual on their chromosome number 2 would have a copy of little r. Little r, they would have two the same, which is possible. Okay, they are separate alleles, however. Okay, now when we go through basically what we call meiosis, we make our sex cells. Remember, when we make our sex cells, we can't give 46 chromosomes, 46 chromosomes to our offspring and have our partner give 46 chromosomes also, because then we would end up with an individual with 92 chromosomes. And that would be problematic. Remember, all of our offspring end up with the same number of chromosomes in all of their cells that we have, 46, 23 pair. So we actually, when we go through making our sex cells, we cut this number in half. And so basically all of our chromosomes get cut in half. So this individual for flower color can either pass on a big R allele to its offspring, or it can pass on a little r allele to its offspring, but not both. A parent will only pass on one copy of each gene to its offspring. Okay? So this parent can pass on either a little r or a little r. Okay? So basically this parent, half of their sex cells are going to contain a little r, and half of their sex cells are going to contain a little r. Now this individual can only pass on little r's. We will still basically write it like so in this case. So the possible offsprings, we'll say this is mom, these are dad's sex cells. The possible offspring are 50% big R, little r, round, 50% little r, wrinkled, or 2 of 4 round, 2 of 4 wrinkled. These are the possible outcomes. Not what will happen for sure, but the possible outcomes. Okay? So on the paper it asks you a question that says, what percentage of the offspring will be round, or should be round? We should change the word there. Should be round, 50%, the 2 in this column. So that's a really easy question, correct? We set up the Punnett square. This parent can possibly pass on that round gene to 50% of its offspring. All right. Now, could it have 8 offspring and they all be round? Yeah, that's possible. Just like some families have 5 daughters, no sons. Okay, it should be 50-50, correct? Doesn't always happen that way, but we're looking at the possible outcome. And from this possible cross, we would expect to see 50% round to 50% wrinkled offspring from that Punnett square. All right, we can do one more. The paper gives us big R crossed with little r. Okay, now at some point in your brain, they might start to kind of go, the light bulb goes off and you're like, I don't have to do that cross, that's obvious, okay? But at this point, we will still do the cross. Okay? At this point, we will still do the cross. So basically, remember, big R, round, little r, wrinkled. This tells us we have a big R individual crossed with a little r individual. Okay, so we set up our Punnett square. We have little r, we will leave that there. But in this case, we have big R times big R. Now this is going to change everything. Okay? This big R times this little r, big R, little r, we're still going to have these two be the same genotype. Big R, little r, big R, little r. Right here, this big R now goes with this little r and this one. So we have those possible outcomes from this Punnett square. All right? So 100% of our offspring would be round with that genotype. Okay, so those are simple Punnett square crosses. Simple. Remember, we talked about the alleles, the chromosomes, how they split in half. We literally write the possible sex chromosomes in the Punnett square and then what the possible offspring genotypes could be. All right? Simple stuff. Next time we'll come up with maybe 2 gene crosses. Okay? Later.